this lecture, that is about the proto-oncogene and about the oncogenes and about the tumor suppressor things. And if you can remember, I have introduced these things in bits in earlier classes. But the terms specifically, the terms about the proto-oncogenes and about the oncogenes and tumor suppressors are important because a cell as a living entity sooner or later will decide to go for proliferation, to go for the cell division. That is the basic and most fundamental property of every cell. Although there are certain cells, if you can remember, that there are certain cells like the neurons, they are not supposed to go for the cell division. They're not supposed to go for, they are programmed not to go for the cell division. But most of the other cells, they are supposed to go for the cell divisions. Now, cell division is a very controlled and a very sophisticated type of cellular activity in which a cell who choose to go for the cell division first think about the need for the cell division, about the job of that cell, whether that cell is designed for the cell division or not, the urgency behind the cell division. So a lot of factors actually defines which help the cells to think whether it should go for the cell division or not. Those factors includes like whether there is enough food out there. That's very important. There is enough food out there. And I believe the very important thing that is there is a need for the cell division. And you can see there are certain type of cells who are in an urgency of cell division, like the skin cells, because they are required to be replaced very often, like the blood cells which are produced in the bone marrow, they are supposed to produce like regularly. So there is a need or not. Enough energy or food is not. After that, after getting all these yes and go ahead signals, the cells plans to go for the cell division. It actually checks itself. During the process, it checks itself whether everything is like fine and everything is going on in a normal way or not. Most of the things we have already studied during that process. If you can remember and recall, the cell choose to duplicate its genetic material, which we call as replication. Cell duplicate its genetic material and then analyze and study whether the duplication is executed in a perfect way or not. Certain mechanism defines and oversee the process of cell division. Those are called as the cell cycle checkpoints. Those are called as cell cycle checkpoints. At a molecular level, these cell cycle checkpoints are controlled by different types of proteins at a molecular level. So the checkpoints and the mechanism which is controlling whether the cell should divide or not, how much a cell should divide or not, is actually controlled by the certain proteins. If this is the protein, then that protein should come from where? It should come from a gene. And if it comes from a gene, that gene is required to be regulated. That gene is required to be regulated. So at the end of the day, whether the cell will go for the cell division or how fast, how often the cell will go for the cell division will depend that how we regulate those genes, how we regulate the genes, which actually in turns regulate the mechanism of cell cycle. Now this thing, if do not controlled, in such a sophisticated manner. If something went wrong during that procedure, you can guess what will happen. This is the normal cell. This is the normal cell. All these are normal cells. And in one of the cell, because of some XYZ reasons, the function of those regulators gone wild, gone mad. There are many types of mistakes. There are many types of dysregulations. There are many ways this mechanism can go out of control. I'm just telling you one of them, that if a cell cycle checkpoint is not functioning normally, it will push the cells to go for the cell division, whether there is a need or not, whether it is required or not, whether there is enough food or not. And that will lead to the large number of those bad cells. Initially, you can see the trouble is less, but since the division is uncontrolled, nobody is checking, nobody is overseeing, nobody is looking at the mechanism perfectly. So with every cell division, we add more troubles, a new type of trouble in the newer cells. Those troubles will kick and push and add new types of troubles. And eventually we have lots of cells with lots of different types of new troubles. And this is what we call as a tumor or cancer cells. So in totality, if the mechanism which is controlling the cell division is gone wild, is gone mad, that can trigger the situation which we call as the cancer. So we should know very accurately that how many different types of genes are out there which are controlling this cell proliferation, cell division mechanism, and how many possible ways are out there which can push them, which can push them to make mistakes. You will see, number one, the most famous, a proto-oncogene, which I have introduced in my earlier classes with you. They are not the bad things by name. The onco stuff is there, but still they are nice. They are important because naturally a cell is supposed to go for the cell division. Somebody should push, somebody should kick the process of cell division, but in a regulated fashion. Those things are called as proto-oncogenes. They can decide and they can control, they can push the cells to go for the cell division. There are different types of proto-oncogenes. Some growth factors are there. Some receptors are proto-oncogenes. Some signal transducers are proto-oncogenes. And many transcription factors can act as a proto-oncogenes. Their presence will help the cells to go for the cell division. For example, G. Eliza, before I proceed.
Sir, मेरा question था कि why do oncogenes exist when they only have to be mutated to turn into cancer-causing genes in the body? Bracha 1, Bracha 2 है, मुझे इनका to be very honest इतना पढ़ के समझ नहीं आता, why do they exist इन्होंने mutate करके cancer ही cause करना?
No, Bracha 1, Bracha 2 के बारे में आपको पता होना चाहिए, I believe, if you study the lecture about DNA repair pathway, the DNA repair mechanism, that why they become like an oncogene. Anything which can lead to the dysregulation, anything which can push the cells to proceed for the cell division without the control, all those things can be considered as an oncogene. Oncogene does not exist over there. Good genes become bad and they can become like an oncogene. Something can push the cells not to go for the cell division at all. It is possible, that something push the cells not to go for the cell division forever. This is either not a very
सर
G.
सर लेकिन ब्राका जीन्स जो होती हैं वो तो ऑलरेडी एक चीज को रेजिस्ट कर रही होती हैं, तो अगर उन्होंने म्यूटेट होके खराब ही होना होता तो फिर व्हाई डू दे एग्जिस्ट इन द फर्स्ट प्लेस?
नहीं, आई थिंक आपको ब्राका जीन के बारे में मुझे ये दोबारा याद करवाना पड़ेगा, या यू आर नॉट वेरी मच एट दिस पर्टिकुलर टाइम यू आर नॉट वेरी मच फेमिलियर अबाउट द ब्राका जीन, जो इसलिए मैंने आपको स्पेशली बताया अगर आपने वो लेक्चर सुना है अच्छे से, you should be able to answer that question, that why BRCA gene become an oncogene. But I am explaining here for you since you have raised that point. Okay, so when the genomic DNA when the cell plans or commit for the cell division, the genomic DNA is supposed to get duplicated. Okay, it's supposed to get duplicated. This is called process of replication. When a DNA got duplicated, there is a high chances that new DNA, the newly synthesized DNA, might contain some mutations, some errors. Those errors can be sensed and can be repaired with the help of the genes. One of them is called as the BRCA1. Okay, they can pick the things and they can fix the errors and they can remove the mutation and convert the gene or convert the DNA into the wild type one once again. So with the help of the BRCA gene, the mutation which arises as a result of the DNA synthesis during the cell division is fixed. Is that clear from here?
Yes.
So what will happen if something bad happen with this the BRCA gene? The BRCA gene is itself produced out of a gene. There should be a gene called the BRCA1, and that gene should be controlled by a promoter region. And that gene, when produced, it will produce a messenger RNA. That messenger RNA will be translated into proteins, and those proteins will execute this function. Now think if a mutation is over there on the BRCA gene. If there is a mutation on the BRCA gene, what will happen? The messenger RNA produced out of the BRCA gene will be mutated. The protein of the BRCA gene will be mutated. That will not going to execute that function. So if the BRCA protein is mutated, the errors which are naturally produced during the process of replication will not be fixed. These mutations and errors will pass on to the next generation. Now the same BRCA gene which is supposed to work and help to fix the mutation is now becoming a reason of letting the mutation go to the next cells. And that will going to cause a lot of troubles, lot of dysfunctions. Let's suppose if the dysfunction is that, if the dysfunction is very big, or if a dysfunction is not accommodable, is not something which a cell can deal, the cell will be dead. And that's might be a better choice because the cell is dead. But what if the mutation and dysregulation will change the dynamics of the cell and cell will start dividing faster? And that is the bad thing. And that is what we call as the cancer. Hope this this explains the question. Okay. So once an oncogene is there, and remember, it does not come from the skies. Our own genes, which are supposed to control the functions, when they gone mad, they can become like an oncogene. And they can remove the controls on the cellular divisions. And that can lead to the trouble. In order to understand a little bit more about your question and about this thought, and I believe the rest of the people are with me in this understanding, we have accelerators in our car. So we have like we have accelerators in our car. The accelerators are supposed to push the cars to speed up to speed the car. So we have accelerators over there. If you ask me, sir, accelerators are the bad things, because if the accelerators will gone wild, will gone mad, that will going to end or result in an accident. Let's suppose if you push the accelerator and it gone wild, you are not able to slow it down, and what will happen? The car will keep moving, keep moving, keep moving, keep moving, and that unfortunately ended up in an accident. So accelerator is a good thing, but when the accelerator is not working in the right way, that will become a reason of an accident. In the same way, think about the brakes. The brakes of the car are the good stuff. I mean, they are supposed to slow down the car when it is required. They are supposed to stop the car when it is required. They are supposed to slow down the speed of the car when it is required accordingly. But if the brakes are not working at a given point of time, they stop doing their jobs, that will going to result in an accident. The brakes are not bad, but they can be a reason of an accident. So similar type of things are present in our cells. So you can imagine that in our cells, there are two types of forces. The one which are pushing the cells to go for the cell division. The other type of forces, they are pushing the cells to stop the cell divisions. The balance between these two defines the outcome, that how much cell division will be there. If the forces which are pushing the cells to go for the cell division, they gone up, out of control, they will going to lead the cancer, uncontrolled cell division. If the forces which are stopping the cells to go for the cell division, if they are not working, eventually the cell will also proceed further for an uncontrolled version of cell division. And that can lead to cancer as well. Interesting question is that, what if those things are not working, who are pushing the cells to go for the cell division? And what about the brakes? If they are working faster, what will happen? You can guess what will happen. The cell will not go anywhere. It will not go for the cell division. And this is also not a natural requirement, because the cells are required to divide after a. If you ask, I will not going to have an accident at all if I stop my car and park my car in my garage. The answer is yes, you will not going to have an accident. But you can also not going to use your car as well. So we need this balance in order to have a regular or natural activity of the cells. So there are two types of things. The one, the oncogenes, the forces which push the cells to go for the cell divisions. When a cell become bad, when a cell become cancer, that depends on these two things, these two phenomena, which I have explained indirectly to you guys. First, that there arise oncogenes, which push the cells to have more cyclins. And cyclins are the factors, proteins, which push the cells to go for the cell division. It helps the cells to jump over the cell cycle checkpoints. You know, the cell is proceed in the form of cell cycle. G, yaad hai? Cell cycle. And there exist certain checkpoints which defines whether the cell will proceed for the cell division or not. Another option, when the cells, we have excess cyclins, we have unstoppable version of cell divisions out there. That will lead to the cancer. If we have excessive inhibitors of P53, P53 is supposed to be like the brakes. It stops the cells to go for the cell division. And if somebody is stopping the P53, that means it will help the cells to proceed for the cell division. The P53 is not there in the system, The cell will choose not to die. The apoptosis will not be there, and that will help the cells to survive. It is not a nice thing to as well. The other thing which can help the cells to develop into tumors or in cancers, if their brakes are not working, if they have not functional tumor suppressor genes. The brakes of the cellular systems, they are called as tumor suppressor genes, which stops and pushes the cells not to go for the cell division. And the rate of the function of these two things define that how much a cell will going to divide. Just for an additional thought, you should understand that once if something bad happens with the cells, for example, I'm just giving you an opening information about the p53. If something bad happens with the cells, if the genomic DNA is broken, mutated, irreversible damage is out there within the cells, and this cell is is no longer a valid cell, is no longer a bona fide cell. It is carrying a very bad mutation or a very, I must say, weird type of, trouble in the genome. It was broken down, or it was like have a lot of mutations. The cells will activate the p53. The p53 will trigger two things. Number one, it will stop the cell to go for the cell division immediately. Stop the cell cycle. You know why? Why to stop the cell cycle? So whatever the errors are here, those errors should not be passed on to the next cells. The errors should not be passed on to the next cell. So it is important that the cells should pause its function for the cell division. And in the meanwhile, allows the DNA repair pathway, which we have studied earlier, to fix the problem. But if the error was not removed, the system stays unstable, then the p53 will trigger another pathway which is called as apoptotic pathway with the help of BCL3 system. That actually changes the permeability of mitochondria. You know, mitochondria has a double membranous layer. It changes the pore size of mitochondria, and that mitochondria will start releasing the components present within the matrix, especially the cytochrome C, especially the cytochrome C. You can see the mitochondria started leaching out, and those cytochrome C will then activate the caspases. Remember, caspases are present within the cells. They are proteases. They can destroy the proteins. Once the caspases got activated, they will start breaking down the proteins present around them, and that will lead to the cell death right from the inside of the cell. And this is called as apoptosis. So that is how cell help the tumor suppressor like p53 help the cell not to pass on the mutations to the next generation. So the next cell should stay like bona fide. They should have a perfect sequence. And if the system fails to fix the error, the p53 push the cells to go to select the death. And it is a very, very unlikely thing a cell will choose, because the ultimate objective of life is to stay alive. But a cell is committed right from the inside to go for a suicide for a greater good. So the bad things should not be passed on to the next generation. Now imagine if this p53 is not working. The errors will be passed on to the next generation. The daughter cells will accumulate errors. They will start behaving weird, and that is a high chance that they will eventually becoming out of control or uncontrolled version of the cells. More easily, they are called as the cancers. Excuse me. So how a proto-oncogene, a good cell, a good thing, a good gene, which is supposed to control the function of the cell division, is becoming bad? So there are two things. The first, either it is a mutation, a point mutation, it can convert a good gene, a proto-oncogene, into an oncogene, or it is a translocation. I'm sure you guys understand these two things. A point mutation, that a proto-oncogene is working nicely, everything is fine, and then something bad happens, a mutation occurs on the gene, and the proto-oncogene will start doing the things in a different way. And translocation is, sometime it happens that one part of the chromosome, this is the chromosome, is actually a full fragment of one of the chromosome during the cell divisions is transferred to another chromosome, is transferred to another, one full part of a chromosome moved and shifted to another chromosome. That will change the locality of the genes present in that portion. For example, here is a proto-oncogene present here. Here is a proto-oncogene present here, and it is controlled in a normal way. But once it is translocated, it moved to another part, and that part is quite active. Nobody is controlling that portion. So what will happen? When that gene is here, it is controlled. When it is translocated, it will become uncontrolled. It will start expressing a lot. And that will going to push the cells to go for uncontrolled cell divisions. A very famous disease called CML, or chronic myeloid leukemia, occurred as a result of this translocation of chromosomal fragments from one chromosome to another. It's quite unfortunate. And that translocation occurs between chromosome number 9 and chromosome number 22. And that lead to a disease, and that disease is also called as Philadelphia chromosome, because it's the newer chromosome has different shape. And that results in the hyperactive of an oncoprotein, hyperstimulation of the cell cycle. And that is what we call as oncogenesis. Those cells will never be able to work in a normal way, and that person will never be able to live in a normal life. There is another way which can push the cells to go for further cell, is amplification. So let's suppose a controller gene or a proto-oncogene has one copy per cell, but due to the different reason, the number of copies within one cell is increased from 1 to, for example, 3 copies per cell. Now the function of the proto-oncogene is same, but since the number of copies are enhanced and increased, the function will be increased. And now the same cell will feel that it is required to express, it is required to proceed for the cell division more often. And let me give you an example of a very famous example of that thing. You can dig the details very easily. There is a receptor called ER receptor present in the breast epithelial cells. Those ER receptors are the proteins present on the surface of the breast epithelial cells. They are supposed to capture the estrogen. Their stimulus is estrogen. When estrogen binds with the estrogen receptor, it triggers a cell signaling cascades, which you should know, and then affects the target genes within the nucleus, and those target genes will push the cell to go for cell division. So in a nutshell, if estrogen is there in the system, that estrogen will go and bind with the estrogen receptor, and the cell will feel and respond in terms of cell division. So the ladies, they experience more estrogen during a one particular time of the month. At that time, they can sense that their breast epithelial cells, they are pushed to go for further cell divisions at that time. Now think about the concept of amplification. Typically, if there is one estrogen receptor present on one cell surface, but because of the mutation, now there are three estrogen receptors are present on these cell surfaces. Now what will happen if we have estrogen in the system? If we have an estrogen in the system, we have such cells which can respond to those estrogens 3 times more than the normal, because they have 3 estrogen receptors expressed on this surface. Now this cell is too much sensitive for the estrogen. If this will proceed with the cell division with a certain speed, with a certain rate, that will going to respond much faster. So such cell will going to have more cell divisions, and that can lead to accumulation of a lot of cells, and this is called breast cancer. When the doctor look at such breast cancer samples, they ask the pathologist to check whether the estrogen receptor is there. They answered like this: sir, this is an ER positive breast cancer. You might remember this term. And that ER positive means that the cells has more than normal estrogen receptors. And that entirely makes sense. Okay? That entirely makes sense. Now, such breast cancers which are ER positive, they are the breast cancer. The molecular reason is the over amplification of an estrogen receptor. And then the last thing is the translocation of a gene from a less active region to an active region. A very famous leukemia, bucket lymphoma, is because of this error, that something is present at a less active region is translocated to an active. I want to talk more on this stuff, okay?